Good evening, everyone. Welcome to our webinar, Diversity and Dementia. I'm Adrian Idenson. I'm the co-director of the UK Dementia Research Institute. Now, the UK Dementia Research Institute is your national institute devoted to understanding what causes dementia and stopping it, and also helping people live safer, better lives with dementia. We're the country's largest dementia research program, with 800 researchers, and we're based in our laboratories in Edinburgh and Cardiff, the universities of Cambridge, King's, Imperial, and University College London. So why, when we're thinking of dementia, do we care about diversity? Well, if you do a Google search right now for images on dementia, you will see lots of media representations of people with dementia. And you will find that almost all of them are elderly. That's not surprising. But almost all of them are white. And almost all of them are sitting in rather nice surroundings that look like very expensive care homes and nice gardens. This suggests they're all very comfortable middle class as well. Now, whereas lots of people with dementia might be like that, did you know that people can get dementia in their 40s, that more women die of dementia than men, and for reasons more complex than women simply living longer, that dementia is more prevalent in Black and South Asian populations than in white populations, and that lower economic status can increase your risk of dementia. The point is that if we're going to be successful treating and preventing dementia, our basic research and the volunteers who take part in our clinical trials and the availability of treatments must take this diversity into account. So this evening, you'll hear from 4 scientists who are not only experts in understanding the biology of dementia, but they are also thinking long and hard about dementia and diversity. So the structure of this evening, we'll have 3 short talks. At any time during the talks, you can look down on the bottom of your screen and click on the Q&A button and type a question, and we'll come to the questions at the end. We'll get to as many of them as possible. After the Q&A session, we're going to show you the world premiere of a very short video about the Institute and some of the other people working on dementia research. The last thing I want to say before we get to the exciting bit is we're recording this, but don't worry, we can't see you or hear you. But if you want to return to this later, we'll give you a link and you can share that with others if you wish. So let's get on with the talks. Our first speaker is Professor Tara Spires-Jones. She's from our Edinburgh Centre, and she's going to talk about the importance of sex and gender in dementia. Tara?
Thank you, Adrian. I hope you're all seeing my slides now. I'm really excited to kick off this webinar on diversity and dementia and how research is bridging the gap. And as Adrian mentioned, I'm going to focus us in first on issues of sex and gender. And I'll talk first a little bit about what I mean by those words. I think it's fairly common sense, but it's good to know what we're talking about together anyway. So gender, I'll refer to as what a person refers to themselves as, what they identify as, whether that's a man or a woman or non-binary. Whereas sex, we talk about as scientists, is a biological trait of humans and animals that's defined by our biology, our genes. And as Adrian says, I've been thinking a lot about this lately. So I'm a neuroscientist working in a lab. We're studying how the brain changes in dementias. And when we do that, we always try and have as diverse a population of donors as we can when we're looking at tissue. And we always look at male and female mice. And I've been thinking about it so much that I wrote this little editorial here, and I'm glad you're going to get a link to this so you can go and read a little bit more detail and some of the references I'll talk about if you're interested. But why are we talking about sex and gender in dementia research? As Adrian mentioned, more women are affected by dementia than men, and it's a lot more, it's 2 to 1. And as Adrian said, some of that is that we tend to live longer, but it is more than that as well. We also know that drugs can work differently in women and men, and there has been historically less representation of women. Women were only included in clinical trials since 1993, and even today, there's some evidence to suggest that women are still underrepresented in clinical trials for brain conditions. And in fact, if you go beyond men and women, people who are transgender or non-binary also have significant health disparities, and there's almost no inclusion of these groups in medical research. So we have some way to go on gender in research. And if we look at why are we focusing on gender and sex and dementia research, we know, and we've known for many years, that male and female brains aren't identical. On average, there are many differences. Also, there's lots of overlap. But there was a study done by some of my colleagues up here in Edinburgh in the psychology department, where they looked at brains from over 5,000 people in the UK Biobank study. And on average, they found that men's brains are a bit bigger, but that women's have thicker neocortex. And that's this beautiful curvy bit on the outside where a bunch of the neurons, the cell bodies, live. And men and women's brains on average also had different connectivity. So it's not hard to imagine that our brains might be more susceptible to diseases of the brain or react differently to drugs to treat these diseases. This is not my area of research, but when I was reading about this for that editorial, I found some really interesting data on how drugs targeting the brain can work differently in men and women. So one pretty striking example was the sleeping pill, zolpidem, that was approved in 1993. But it was found very quickly that when it was started being given to people, that blood levels of the drug were much higher in women than men, and that women were having impaired driving the next day, even though they were taking the recommended dose. So this eventually caused changes in the dose for women, at least in the States. And there's been some really interesting biology since then to show that this is due to a difference in the chemicals in the brain that clear the drug between men and women. So it's just very important. If we want dementia drugs to work for everyone, we need to make sure that we include everyone in the basic research. And then if we talk about even more fundamental research, stepping back from clinical trials and men and women's brains, and we think about the fundamental neuroscience, which is how we in the UK Dementia Research Institute and many others around the world are discovering these really important brain changes that we're then trying to target and eventually get to drugs in people. Well, historically, this kind of study that has been done in animal models, for example, or cell lines, this data I'm showing you is from animal rodent models, and historically people only used male mice. And the logic behind this was that they thought that because of the estrous cycle, female mice would be more variable, and it's also cheaper to use one sex of mouse than two sexes of mice. This is what the data looked like in about, I can't remember when this was 2018, where about half of neuroscience studies were using only male mice, and some studies didn't even specify what kind of mice they were using. But more recently, this idea that male mice are less variable than females has been challenged, and there's been a huge amount of data and a big meta-analysis, which is some scientists that looked at huge numbers of studies that have examined this. They looked at over 26,000 mice in this meta-analysis, and they actually found that the sex bias in how different the mice were was really dependent, with some traits being more variable in male mice and some of the traits being more variable in females. So we're moving now as fundamental neuroscientists to the understanding that we need to use both sexes of mice because some of those biological traits are shared between mice and humans. So moving forward, we are having some new policies. So the Medical Research Council that funds part of the UK Dementia Research Institute and much broader neuroscience around the country has now mandated that when we do our fundamental neuroscience, we look at both sexes, and they're willing to fund it, which is important for our work. There are also regulations now from the European Medicines Agency and the FDA in the United States, which have more rules about you need to include populations that are more representative, not just men and women, but as we'll hear about a little bit later, people from different racial backgrounds and lots of different types of diversity that we need to include in these studies. And there's also been quite a lot of grassroots initiatives coming from scientists. So we formed in the UK Dementia Research Institute a diversity think tank, the scientists coming together thinking, what can we do about this problem? We're also doing it on the level of journal publishing. We're thinking about the representation of this actual scientists and our diversity within our field as people who are contributing the ideas, because we need the most diverse minds as well to solve these complex problems of dementia. So finally, oh, sorry, my slide went forward there. So finally, I just wanted to end on a positive note. So fundamental research, like we do throughout the UK DRI, really works. And I'm showing you one example of fundamental neuroscience research that went on to help people here. So this is probably one of my favorite science news stories of the past decade. And this is not someone with dementia. Obviously, this little guy here is a little bit young for even early onset dementia, but he was born with something called spinal muscular atrophy, which is a type of motor neuron disease. And just a few years ago, this was a horrific, and still is a very horrific disease, but a few years ago there was nothing to be done about it. And most of these kids who have this disease would die very early on in life. But because of really fundamental research into genetics and neuroscience, there was a treatment approved a few years ago now, and now these kids are living and meeting motor milestones. So that's an example of how you can take neuroscience and make it into something that works. And closer to our hearts as dementia researchers, there was some news recently that a clinical trial looks like, we haven't seen the data yet, but it looks like there is a new treatment that can slow cognitive decline in dementia. And again, that's due to basic neuroscience research translating. And what we want to focus on here in this last couple of minutes in our webinar today is we want to be sure that these kind of amazing things, these treatments for SMA or these new treatments for dementias, are going to be relevant and help everybody who ends up living with dementia, not just, as Adrian mentioned, a subset of people who participate in our studies because they have the opportunity to do so. So I'll finish there, and I look forward to the Q&A, and I'll try and stop my screen. Thanks, everyone.
Thanks so much, Tara. That's great, and I love the fact that you're ending on a positive note. It is not all doom and gloom, and in fact, the field of dementia research is sort of on fire and achieving a lot these days. So great, thank you, Tara. Okay, our next speaker is Dr. Frances Weisman. Frances is a group leader, a researcher here at the DRI's UCL London-based labs. And she's going to talk to us on something that's going to surprise you a little bit in terms of dementia. She's going to talk to us about Down syndrome and dementia. So, Frances.
Thanks so much, Adrian. Thanks so much for the opportunity to talk to you this evening, and thanks everyone online for their time. So as Adrian said, my name is Frances Weisman, and my group at the DRI understand the development of Alzheimer's disease dementia in people who have Down syndrome. So I'm just going to explain to you a little bit, for those of you who are in the audience who aren't familiar, what Down syndrome is. So Down syndrome is a genetic condition, and about 1 in 1,000 babies born have the condition, and it affects people differently. Now, we know the condition is caused by having an extra chromosome, and chromosomes can be thought of as chapters in our DNA instruction book. So DNA and those instructions tell our cells and our bodies how to function. And in typically developing population, people inherit pairs of chromosomes, so one pair from their mum, one from their dad. But in individuals with Down syndrome, they actually inherit an extra chromosome, so chromosome 21, which is one of the smallest chromosomes. So all we know about the Down syndrome is that everything associated with the condition is caused by having an extra copy of the genes that are encoded on this chromosome 21. So we know that people with Down syndrome have an increased risk of developing a number of medical conditions. So in my group and other people within the DRI work on the development of Alzheimer's disease, the early development of Alzheimer's disease, which is a form of dementia in individuals with Down syndrome. The individuals with Down syndrome also have got an increased risk of developing epilepsy, childhood leukemias, autoimmune disorders, and obstructive sleep apnea. But they have a decreased risk of developing some other conditions, so cancer, solid tumours, reduced risk of developing high blood pressure and coronary artery disease. And we know that all these changes in the risk of these medical conditions are caused by having that extra copy of that chromosome. So many of you may have heard in the news that individuals with Down syndrome were quite greatly affected by the COVID-19 pandemic. And this is one of the studies that actually came out first from researchers at the University of Oxford, who are looking to what happened to people with Down syndrome after they were hospitalized for a COVID-19 infection. So those are the people that you can see in the red bars, and the individual matched people from the general population are in the blue bars. So if you look at the blue bars, we've got the oldest people here on the left and the youngest people on the right. We can see that age-associated risk of COVID-19 mortality risk that many of us have heard about. But you can also see that really striking increase in those red bars about how much higher the risk of dying from COVID-19 is for people who have Down syndrome. So a number of people have been researching that, not my own lab, but in the wider Down syndrome research community. And we think some of those are because of biological changes, because of having an extra copy of chromosome 21. But colleagues of mine, the London Down Syndrome Consortium, which comes out of King's, have actually been looking how access to healthcare influence that risk of mortality. And in this study by Assad, which he published just this year, they demonstrated that a part of the increase in risk of mortality mortality of individuals who had intellectual disability or ID was actually because of impaired healthcare access. So once an individual who had ID was admitted to hospital, they were less likely to be admitted into ICU than matched cases who didn't have intellectual disability, and they were less likely to have other interventions like intubation. And this may have contributed to the higher mortality in those individuals that we saw during particularly the first and second waves of the pandemic. So it's very important that we remember that we need to increase access to healthcare, so existing healthcare for people with ID, including those with Down syndrome, but we also need to understand the medical needs of those individuals. And this is where undertaking fundamental research to address the needs of people who have intellectual disability comes in, and this is how we're addressing it in the DRI. So my group are trying to understand why it is that people who have Down syndrome have such a high risk of developing early onset Alzheimer's disease. So we know that that half of people who have Down syndrome will have a clinical dementia diagnosis by the age of 52. So that's current European studies suggest that. And that we also know from work led by Andre a few years ago that dementia is the leading cause of mortality for adults in the UK who have Down syndrome. But we also know, because we know that those increased risks are caused by just having an extra copy of the chromosome, the chromosome 21, and we know what that chromosome is, and we actually know that it's the smallest human autosome, so one of the smallest chromosomes, and it actually only contains about 1% of the coding genes of the human genome. So that gives us a great big clue to understand how that we can start to unpick why it is that people who have Down syndrome develop early onset Alzheimer's disease. So my lab are trying to work which of those genes is important. So before I joined working in this area, we actually knew that one gene on the chromosome called APP, or amyloid precursor protein, is super important for the development of Alzheimer's disease in individuals with Down syndrome. And the reason we know this gene is so important is because some very special people with Down syndrome that were observed by clinical colleagues of mine, including V. Pressure in Birmingham and also Ira Lott in the US, who were very elderly individuals who had Down syndrome, who were completely cognitively fine. They hadn't developed dementia, and they hadn't developed any of the other features of Alzheimer's disease we can see by brain imaging studies. And when the genetics of those people was looked at, it was noted that one of the genes on the chromosome, or some of the genes on the chromosome that was missing in those individuals that they didn't have an extra copy of, was this gene called APP. And we also know from work by many colleagues in the field that the APP gene actually encodes a protein, which is the protein which forms the plaques that accumulate in the brains of individuals with Alzheimer's disease, whether they do or don't have Down syndrome. But we also know that Alzheimer's disease is a super complicated process. It's a cascade of a lot of changes in the brain, which including this accumulation of the plaques. And we also know from my work of myself and a number of other colleagues that genes on chromosome 21, and this is this very complicated figure, which is just the take-home message there is it's complicated. We know that, that the genes on chromosome 21, when you have an extra copy of them, as people with Down syndrome do, that changes aspects of how that complicated cascade and all those changes that in the brain that lead to individuals to develop dementia are affected. So we've been trying to understand how the pathways are similar and how they're different in people with Down syndrome. And that's really important when we start to think about translating to clinical trials, which we're heavily involved in, especially Andre's team, because when we take a drug to clinical trial for individuals with Down syndrome, we need to make sure that it's going to work in the way that we think it is, and taking data just from the general population may not be, give us a safe and effective treatment for this really important group of individuals. So in my group, we tackle this in three main ways. So we study brain samples from people who both had and did not have Down syndrome, and those samples were donated to brain banks after the people died, generously shared with scientists, so that we can look at them and understand how those brains differ between people who had the extra copy of the chromosome and didn't. And we can also look at cellular models or animal models which have an extra copy of the whole chromosome or genes from the chromosome, and understand how those pathways differ. So it just remains for me to thank my current members of my team, so Yijing, Paige, Cleona and Kat, all the London Down syndrome participants and the brain bank donors and their families for facilitating our research. And if you're interested in any of the work that you've heard today, or have a family member who has Down syndrome who might be interested in participating in clinical research, please do contact the clinical team at downsyndrome@kcl.ac.uk, or check us out on Twitter if you want to see what we've been up to, or just email me. Thanks very much.
Thanks, Frances. That's terrific. Yeah, we don't often associate younger people with intellectual disabilities with dementia. So I think that's really quite enlightening. Just before we move on, I want to remind you that down on the bottom of your screen is a button Q&A. If you'd like to ask any of our speakers a question, or even a general question, then click on that and type it in, and we'll get your question. There are a few building up there, so please take advantage of that. Okay, we're going to move on to our third and final talk. This is a bit of a double act with two colleagues here at UCL. Dr. Mee Ridzik is with the Institute of Neurology here at UCL, and her junior assistant, Professor Sir John Hardy, who's sitting to my left, is also a group leader. And I think, John, you're going to start us off and then pass it over to me.
Thanks for attending this. Obviously, this is thanks for this. Just got a message that I had not unmuted myself. Sorry about that. So as Adrian said, of course, the idea that we have as an institute is we want to help understand all the neurodegenerative diseases across the world, in all races. And I think it's useful to start with this diagram, which shamefully I took from the Daily Mail, and that is the history of humanity. And the history of humanity is about 100,000 years ago, a group of people, quite a small group of people, left Africa, probably via the Gulf here, and then a group turned left and came to Europe, and another group turned right. Well, actually two other groups. One group went across down to Australia. At that time, the sea level was 50 feet lower, and it was much more of a land route than it would be today. And then another group came through Asia and down through the Americas. Now, why is this important? Well, it's important because the group that left Africa was quite small, probably something of the order of 50 to 100,000 people. So that means that the genetic variability in all of these individuals throughout the world is tiny in comparison to the huge genetic variability in Africa. And that means that, you know, if we want to understand the risk genes for any disease, the place to look is Africa, because we have far greater genetic diversity. So far, nearly all of the genetic studies, it's something, I think Mee's going to give you the figures, but it's something like 90% of the genetic analysis of common neurodegenerative diseases, like Alzheimer's disease and Parkinson's disease, have been carried out in Europeans. A substantial set have been carried out in China and Japan, but almost none have happened in Africa. And as I've said, that's a very important point. Now, one of the facts we've learned about all the neurodegenerative diseases is we want to get better at treating the diseases earlier. So we want to get better at predicting who is going to get the disease earlier. And so obviously the other side of this is we need to be better at predicting the disease across the world, and especially in Africans. And so it's of benefit to us as Europeans, but of course it's a moral imperative for us to understand the diseases across the world. And it's been my great pleasure to work with Mee Ridzik, who has been setting up these projects, largely in Parkinson's disease, with colleagues, particularly in Nigeria, but in other African countries, to try and understand the disease in African populations. Mee, over to you, love.
So thank you very much, John, and I'm very delighted to be with you here today to talk to you about this initiative, which started at Queen Square about 4 years ago. I'm just trying to adjust my slide here, so just to go to the next slide. So if you can give me just a second, I will be able to move. So I'm a neurologist and I'm a molecular geneticist. I'm an African as well. I grew in Africa. I lived part of my adult life in the UK and I work with colleagues in different settings. So this particular project is very, very close to my heart. It started about 4 years ago in Queen Square in collaboration with multiple institutions in Africa. And in the NIH in America. And you will see from my slides that it's a kind of a global effort and is trying to increase diversity and include different ethnic groups and population in research in neurodegeneration. The consortium, which I will be talking to you about today, is called the International Parkinson's Disease Genomic Consortium. It focuses on Parkinson's disease, but it will cover other dementias related to Parkinson's disease and other diseases as well. To cover the definition of health inequalities, and I think that we've heard excellent examples about disparities in health and health inequalities from the previous speakers, but health inequalities definition is the unfair and the systematic lack of inequalities in access to care due to factors such as socioeconomic factors, specific ethnic characteristics, or sexual preferences, or even the locations of the individuals. And sometimes these inequalities are amplified in people who have more than one type of these advantages. And the typical example of this is disparities in genetic research in African population because of multiple factors, including socioeconomic factors, income factors, the location and geographical distribution of these individuals. And I looked yesterday at a particular website which is calculating the number of participants in genome-wide association studies. These are the studies which looks at the association of certain genetic mutations of variants with disease. And it breaks the number of individuals who are participating in these studies by ethnicities. And you could see the stark differences based on ethnicities. Africans are disproportionately represented in these, underrepresented in these studies. About 0.3% of the number of individuals who are included in GWAS studies are from an African-American, or Afro-Caribbean, in comparison to about 95% from a European background. Obviously, this is clearly harmful, and this is why we're here and why we're talking about it. And when it comes into neurodegeneration, is specifically relevant, because when we look at the percentage of individuals who are affected by neurodegeneration, including dementia, Black people are approximately twice as likely than white people to be affected by Alzheimer's disease. And in a similar way, Parkinson's disease have a similar overrepresentation in people from a Black or African background. These are data coming from the Global Burden of Disease, which is a global study calculating the prevalence of diseases across the world. And what we know from the latest figure from these studies, that Parkinson's disease prevalence is as twice as in Africa in comparison to Europe and America. Besides that, we know that large population, large number of people will come with neurodegenerative diseases, will develop neurodegenerative diseases by the mid of this century. And there are two contributing factors to this. The population of Africa is increasing, and it will be 1/4 of the world population by the year 2050. And people within the African continent are living longer. And these two factors together will cause rising in numbers of people with neurodegeneration from these population. And this will be a significant burden socially and economically, and this will be globally, and is affecting everyone around the world. And as John said, there is a missed opportunity by not involving people from an African ancestries in genetic studies. And the reason for this, because there is huge genetic variability within the African populations. And harnessing these genetic variability will not just help the African only, it will help all nations, because it will help us identify genetic mutation easily, and it will help us identify pathways leading to diseases, and would help us devise diagnostic and therapeutic tools quicker. So we can argue that by not involving Africans with diverse population, we are actually stymieing the progress in science. And the bigger argument is it's the right thing to do. It's a moral and it's an ethical question, and it's very important to provide treatment, access to care and therapy to everyone, regardless of their ethnic background. So this will bring me to the work which we started about 4 years ago. I'm very, very proud to be coordinating this and working with my colleagues from around Africa and different institutions in Europe and America. It's the International Parkinson's Disease Genomic Consortium is a pan-African consortium. We're focusing on understanding Parkinson's disease and the other dementia. We're focusing on clinical and genetic research, education and training, capacity building, and at the same time, public involvement. We're blessed by being funded by multiple organizations. They believe in our mission and they're helping us achieving our target, including the Michael J. Fox Foundation, several programs, including the Global Parkinson Genetic Program, which is an international program supporting the investigation of genetic backgrounds of about 150,000 individuals across the world from different ethnicities. And we're receiving funding from the Grand Challenges as well. As I said, we do have multiple collaborators from around the world, and this map is showing our collaborators from Asia, Latin America, and Europe. We are now about 110 collaborators in 15 institutions and 12 countries. I've listed them here, but I don't have the time to go through them. For individuals who are interested to know more about our consortium, and I encourage people actually to read this correspondence in the Lancet describing our mission, our initiative, and the contributors to the research. As I said, our deliverable is to build a platform which will enhance the diversity of Parkinson's disease genetics and other dementias. We share our mission with a lot of other platforms, and our data is available for people to share. And at the same time, we are investing in diversifying researchers by investing in education and training, and we're reaching to the patients and the public. I think within the last 4 years, we achieved quite a lot. We managed to publish a few papers coming particularly from Nigeria. We managed to replicate a few results from the Europeans, and we are about to publish the first GWAS study in Parkinson's disease in Black population. And this is kind of, this will show that through collaboration and goodwill, people can achieve significant advance within a shorter space of time. At the same time, we're looking at environmental risk factors which might contribute to the disease. And this is something which you cannot do in a high income setting. So because a lot of air pollutions and heat and metals and other pollutants could be accessed and could be analyzed in a better setting in low and middle income countries. As I said, we're not just diversifying the data, and we're not only diversifying the participants to the research, we're diversifying the researchers as well. And this is quite important. You cannot conduct a research relevant to patients without diversifying the group of individuals who are studying the disease itself. So we do have a huge network of people who are investing in education and training and skilling people in Africa and our cross networks. In addition, we're invested quite a lot in patients and public involvement. So this is particularly, this particular initiative, I'm really very proud of, because we identified the barriers which were stopping us from interacting with individuals. And the most important barrier was the language. So we invested on building up a campaign, which is a health literacy campaign, to translate materials in about 11 African languages, is spoken by about 600 million individuals. And the language is kind of, it's been displayed in this slide, is Swahili, Arabic, French, Pidgin, Hausa, Yoruba, all these are local languages. And we build booklets and podcasts for those individuals. These materials have been generated by experts and edited by patients themselves and their carers, and we use culturally sensitive language and appropriate messaging. And you can't do research without engaging with the local communities. So we're being very, very careful from the beginning to reach to people within the community and make the research question relevant to them, so we can feed back this information to them. And these are images from the participation of patients and the carer in our World Parkinson Day this year from Nigeria, Ghana, and Cameroon. So what are our future directions? Our future direction will continue working in Africa. We will continue producing data, and hopefully we'll share our data with the international community. But because I'm presenting here in the UK, we know that we are deficient in diversifying data in the UK as well. So we're hoping that by next year, we will launch a study to look at the Black population within London, with the hope that we can capture and diversify the data from the population within the UK as well. So I need to thank my collaborators. I have a list of about 150 collaborators worked with us in this project, and we are actively looking for collaborators, and I'm looking forward actually to receive your questions and feedback. And thank you very much for giving me the opportunity to be with you here today.
Thanks so much, Mee. That was terrific. We learned a lot, human evolution movement and research in sub-Saharan and North Africa. That's great. I'm going to ask the speakers to please put your cameras on, but keep yourself muted until you're talking. And we're going to dive into the questions and answers. So thanks to all those who are submitting questions. Keep them coming. And we're going to start off with a couple of questions on lecanemab. So I'm going to turn this over to John and then Tara, just take fair warning. And John, maybe you can start off by explaining to us what sort of drug is lecanemab and why is everyone talking about it at the moment?
So in the brains of Alzheimer patients, you get a protein called amyloid, and lecanemab is an antibody which basically sucks the amyloid out of the brain. That's a simple-minded mechanism. And other drugs have been tried in this area before, I think, and this one looks as if it's been successful. I think the main reason, in my view, it's been successful is because the clinical trial was really aimed at people who were very early in the disease process. And I think that there might be other reasons as well, but I think that's a major reason. And that plays into our talks tonight, because what we really need to be able to do to give this drug in other than to wealthy people is to get better at diagnosing the disease early in other, well, even here in the UK, We need to get much better, but you can imagine it's very difficult to diagnose the disease very early, for example, in African populations. So this is great news, but it really means we need to get better at diagnosing, and that's one of the really impetuses behind the type of work that ME is doing. We also, of course, and I saw one question about this, need to deal with the fact that these drugs are of their nature going to be expensive. The research has been expensive for the drug company to develop this drug, and so the prices are going to be high, and that also is going to be a pressure that we have to, as a society, deal with. So this is great news, but it poses difficult questions for the next stage to get it into people.
Thanks, John. And just before I turn to Tara, are you aware that this trial, how did they approach the question of diversity? I mean, ESI, who sponsored the trial, have said that they paid particular attention to this in the volunteers that are used in the trial.
I've heard that too. I don't know the answer to that. I do know that because I actually have spoken to ISI scientists, that they were very proud of involving African Americans, for example, in the trial. I know that, but I don't know the details. We're of course waiting. What we've seen is a press release. We're waiting for the full paper to be published, and the full data are going to be released at the end of November. So I'll be able to answer that question better at the end of November.
Great, thanks, John. I do recall them saying that up to 25% of the people in the trial were from African American or Hispanic background, and that's unusual. I'm going to move over to Tara now. Tara, would you like to address this question that from one of our other attendees this evening, that was really talking about more broadly, not just lecanemab, but health inequalities? John touched on this a little bit, but unpack that a bit for us. How does a drug like this, where is it likely to be used first? And will it increase health inequalities or decrease them in the field of dementia?
Well, I'll start off by saying this is not my core field of expertise, not being a clinician or involved in the clinical trials, but from my knowledge of the field, what John has just talked about with the cost is going to be a big factor. We know that this, not only was it expensive to develop, but this has to be infused in your blood, I believe, every month, and it's very expensive. So that might increase inequities if it's not covered by governments, like if it's not on the NHS or Medicare in the United States. But more broadly, these drugs will have that problem and just getting access. There was also a nice question in the chat about the sort of question of what are these health disparities, and one was about where you live. And that's something that's quite interesting, and Mia covered as well. Where you live does have an impact in terms of your risk for dementias. And it's not really biologically clear exactly why that is now, but there's a lot in there that we are starting to understand brain links. For example, education, and more education is protective, and you have a less chance of developing dementia, but a lot of other things about coming from a place with low socioeconomic status, and places with pollution, and places with sort of less opportunity, where people have less healthy eating habits, for example, is associated with increased risk. And those are all the sorts of things we need to start to think about and unpick, and really to make a difference to everyone living with dementia. I'll end with a little plug for something up here in Scotland. I don't know how many of our attendees are up here, but one of a group up here, Alzheimer's Scotland, and some of my colleagues have started brain health clinics specifically targeted to neighborhoods where these risk factors are all high. So people can come in and talk about their brain health and their dementia risk and try and modify them before they end up with the disease.
Thanks, Tara. Terrific. I'm going to turn now to Francis. So Francis, this is a question for you. Have patients with Down syndrome been included in any of these clinical trials aimed at dementia?
So People with Down syndrome are usually specifically excluded from inclusion in AD clinical trials, and that's because of their pre-existing intellectual disability and that the same cognitive assessments can't be used. So there's an international consortium of researchers, Horizon 21, led by Andre here in the UK, so that's the European arm, and also in the US, and they have been working for trials specifically for people who have Down syndrome, so developing all the measurements and tools that they need, the cognitive assessment tools. And they have now a clinical trial ready cohort, and there have been some early clinical trials. So there's one by a company called AC Immune in the US, which uses drugs that are similar but not identical to the ones that we've been discussing earlier. And I know that there are talks going on at the moment about repeating some of the more recent trials in individuals with Down syndrome to see if the same effects, but they weren't included in the main clinical trials that we've been reported about recently in the media. Thanks.
Thanks, Frances. Mia, I've got one for you. So Dimitrios says, I've got an objection. In Africa, we do not have high percentages of dementia because people's lifespan is shorter for various reasons. The longer you live, the more likely you are to get dementia, hence why Europe and Asia have higher prevalence. So I think in your presentation, you were talking about cases, and you might want to unpack because there's some truth to what Dimitrios says here, but you can address that, I think.
Yeah. So it's quite a complex questions, because the first thing is you need to have the right set of prevalence. So you need to have a good epidemiology study to understand the number of individuals who have been affected by the disease through their lifespan. And at the moment, unfortunately, in Africa, we do not have the full figure from everywhere. And this is kind of affecting our ability to estimate the prevalence of diseases. So the majority of the studies where we understood the prevalence and the number of people who are affected were estimated prevalence. And this is based on databases, which we've collected randomly, and we made projections. And this is really hard. So at the moment, we do not have true prevalence for any one of the neurodegenerative condition, whether dementia, whether Parkinson's disease, and so on and so forth. So this is the first difficulty, the lack of data. The second problem is we don't have sufficient research on the genetic factors themselves. So if we start to understand the biology of the disease, we need to investigate what are the risk factors for people to develop them. So from my observation in the study which we conducted, and I can just speak for Parkinson's disease, not for Alzheimer's disease, we've realized that people develop Parkinson's early in Africa. So we do have a higher percentage of people who have severe phenotypes, this is severe disease, and early in life. So there are a lot of genetic risk factors which we didn't understand and we didn't kind of investigate yet. And there are other environmental risk factors, including pollutants, which we are addressing at the moment, and diet, and so on and so forth. So it's very, very early to say why people develop dementia and at which time in their life. And certainly we don't have enough figure and statistic for us to have a true estimate. And from observation only, we can say people perhaps they develop lower, they are lower prevalence, but is not the true prevalence for the disease.
Thanks, Me. It's complicated, isn't it? And we've made a start, but we've got some way to go on these questions. Thanks for the question,
Actually,
Demetrios.
worth saying, sorry to butt in, but the African population is aging faster than any other population. So the population increase in the elderly Africans is going up a lot right now, much more, for example, than it is here in the UK. Correct me if I'm wrong about that, but that's my understanding.
I think it's absolutely correct, because if you can think about the number of people and the population in Africa now is growing quickly. So in 30 years, the number, and they're living longer. they're definitely living on longer, and the living standards are getting better. So the socioeconomic circumstances, which all the infectious diseases which are causing early death, is definitely now they are less than they were about 30, 40 years ago. So, and they're blooming economies in Nigeria, in North Africa, in Egypt. So at some point in 2060 or 2070, definitely the people who will be above 65 will be more than the people who are living in Europe. And we're expecting that a significant number of those individuals will be affected by neurodegenerative conditions.
Yeah, good. Thanks, John. Thanks, me. Yeah, absolutely. That's something we all have to look out for. Tara, I'm going to move back up to back north of the border. And ask you a question about, is there evidence for dementia risk being different depending on where you live in the UK?
Yes, and that circles back to a little bit of what I was trying to say earlier. Your dementia risk does depend on where you live. It's not so much about Scotland or England or Wales, but it's about your actual neighborhood and your local environment and how many advantages you have there in terms of low pollution and in terms of good education and lots of opportunities. So yes, it does, your risk does vary. And like I say, we don't fully understand all of the reasons for that, how these biological risk factors change your brain, but a few things that seem to be protective are education. So this is great, everybody. Well done for coming and listening for an hour in the evening. Keeping socially active is very good. Keeping physically active, anything that's good for your heart, seems to be very good. And the opportunities to do this just seem to vary, and your diet and things and the availability of good food and nutritious food varies based on where you live. And things to avoid are things like head injury, like when I fell off a horse this year, that was not good. Things like too much drinking and too much smoking, things that you would already associate with poor health, we now know are also associated very strongly in some cases with your risk for dementia. So it's not to blame people who have dementia, because it's only about 1/3 to 40% of dementias that we think are accounted for by these modifiable risk factors. And a lot of people, as me and John have been talking about, have a combination of unlucky genes. But at the same time, that we can all do our best to reduce our risk a bit, and hopefully we'll have lucky enough genes. And if we live a healthy enough lifestyle, we can avoid this altogether.
Thanks, Tara. That's going to be a quote for tomorrow. Don't drink and smoke whilst horse riding. It's really not good for your brain. Okay, I'm going to move what will probably be our last question. Maybe we'll squeeze another one in, but I'm going to move to Frances, and I'm going to ask her, what would be your top tip for researchers to ensure that their research is inclusive? What measures can be taken?
I think taking a step back and thinking about how your research is in the bigger picture. And that's kind of how I came to be involved in the study of intellectual disability. So thinking about actually the people that are affected by the condition you're studying, meeting them, speaking with them, taking the opportunity to have conversations and understanding what affects them. I think that's one of the major important things for us to include and think about diversity in our research, and then how to implement that knowledge that we have from people that have the conditions into our research to make our research relevant and important for their clinical needs. I think those are the key things to actually scientifically address, so that we can make sure that the people that are affected are, that our fundamental research is addressing their real needs.
Great, thank you, Frances. I'm going to squeeze one last one in, and I'm going to go back north to you, Tara. You've touched on this a little bit, so I wonder if you could take a minute or two to sort of flesh this out a bit. But a few of the questions are really aiming at the same basic issue, and that is, can we often see things published in the media about, you know, this is good for your brain and this is good for your brain and this will help you, and Sudoku and music and what have you. Talk to us a little bit about, is there evidence that people can change their lifestyle to reduce risk? And if so, what sort of scale of risk reduction can we consider?
That's a great question. And as many questions in science, there's not a very satisfying and complete answer yet, I would say. But there is some evidence that changing lifestyle factors has reduced dementia risk on a population level. So there's some really cool data from Carol Brain and her group showing that even though the number of people with dementia is going up because our population is aging and age is the biggest risk factor, if you look at within a certain population, like each individual person's risk, that's actually slightly gone down over the past few years in places like the UK where we have good healthcare. And we don't fully understand why that is, but the best thoughts and evidence are that it's things like improving cardiovascular health, which we know is important for brain health, has actually led to decreased risk. So there's a little bit of epidemiological evidence, which can't tell you causation or really biological mechanisms, but it's really cool and promising. And there have been a few trials trying to change people's lifestyle, intervening to see if you can prevent dementia. There's something in Finland, the finger trial, where people were given an exercise intervention and then were followed up to see if they developed dementia or not. And I would say the data so far have not been phenomenally, overpoweringly convincing yet, but I think it's going to take a lot of time. These are diseases that develop over decades. That amyloid that John was telling us about builds up in the brain for 20 years before you have symptoms. So if you want to do a lifestyle intervention trial, you really need to start looking in midlife and follow people up for decades. So evidence isn't super solid yet, but I would say very promising, and it's good for you anyway to do this kind of thing. So it's not going to hurt us all to do our best.
Great, Tara, thanks so much. Okay, we're going to, that's the end of the Q&A. We're coming up to the hour and we've got a couple of final important things to do. So first of all, thank you, Tara, thank you, Francis, thank you, John, thank you, me, for really interesting presentations there. Thank you to all of you who joined us this evening. As Tara said, you're doing the right thing, keeping cognitively active at this time in the evening. Well done. But thank you also for all your questions. That was really interesting. Tomorrow you'll get an email from us. Two purposes to it. First of all, entirely up to you, but if you like, you can use that email to sign up to our occasional communications and mailing list. Entirely up to you, but if you'd like to hear a little bit about what's happening in the Institute and we'll send you the occasional article, then that'll give you an opportunity to sign up. The other thing is very important. We'll ask you for some feedback. We want to get better at talking to the broader community about our research. So give us some feedback, tell us what we got right, tell us what we got wrong, tell us what you'd like us to do next time, and we will absolutely take that into account. With that, I'm going to end by turning the screen over to our world premiere. It's a very short 2-minute video, tells you a little bit more about the DRI and our work and some of the other people who are doing it. Thank you all very much and have a great evening. Bye-bye.
So to make it really personal and to be really honest, I think he had a real hard time. He never expressed to me how he felt. However, he did tell my mom that he thinks he might be in trouble.
Dementia is one of the greatest challenges facing the healthcare system in the UK. We have an ageing population. One of the main risk factors of dementia is age. 1 in 4 hospital beds are taken up by someone with dementia. The UK DRI was set up to tackle dementia as this great scientific challenge. All these dementia researchers tackling the problem from multiple angles.
I'm Dane. I'm looking at the earliest changes that happen in Parkinson's disease to try and understand how we can combat the disease at the earliest stages.
Well, I'm Shlomi Har. I'm a motor neuroscientist. We're developing objective digital biomarkers for the disease progression that can serve as a benchmark for any future therapy for people living with dementia today and in the future.
My name is Kitty. My main project is to develop non-pharmacological and non-invasive treatments for Alzheimer's disease.
I study the immune system of the brain. I study how the immune cells help to protect the brain as well as help to maintain the proper function of the brain. And even though it's too late for my dad, we will bring a cure that will not be too late for other families.